All right, everyone, welcome to the deep dive. I'm actually hosting today because Arav is late.
सॉरी राजीव चौक से मेट्रो मिस हो गई.
भाई तू तो हमेशा लेट है. ट्रांसलेशन की तरह डिलेड प्रोसेस.
Nice segue, Utsav. Actually, that's perfect because translation is a multi step process. And today we are breaking down how your cells make proteins from mRNA.
Ma'am, I've noticed NEET questions on translation have become super specific. They're asking about initiation factors, elongation cycles, even the wobble hypothesis now.
Exactly, because translation is where the genetic code actually gets read. DNA makes RNA, fine, but translation? This is where RNA becomes protein. This is where life actually happens.
तो बेसिकली ये बायोलॉजी का मैन्युफैक्चरिंग यूनिट है.
Perfect analogy. Think of it like the Maruti Suzuki plant in Manesar. You have blueprints, mRNA, raw materials, amino acids, assembly line workers, tRNA, the actual factory floor, ribosome, and quality control, release factors. सब कुछ organized है.
अरे मानेसर प्लांट. भाई मैंने एक बार फैक्ट्री टूर किया था स्कूल से. एकदम असेंबली लाइन था. एक जगह स्टीयरिंग व्हील, दूसरी जगह टायर्स, तीसरी जगह पेंटिंग.
That's actually a great analogy for polyribosomes. Multiple ribosomes on one mRNA strand, each making one protein copy. Mass production.
Exactly. But DW में क्या होता है? They'll show you one diagram of a ribosome and say, ये translation करता है. Cool. But how does it know where to start? Where to stop? How does tRNA find the right amino acid?
और दून अकेडमी, भाई उन्होंने तो एनिमेटेड फैक्ट्री बनाया था. अमीनो एसिड मैन डिलीवर्स पैकेजेस.
यार ये अमीनो एसिड मैन कौन है? सलमान खान का नया कैरेक्टर.
Stop it. Okay, listen. Translation has three main phases: initiation, elongation, and termination. Think of it like ordering from Swiggy. Initiation equals placing the order. Elongation equals food being prepared step by step. Termination equals delivery complete, app closes.
That's actually helpful for remembering the sequence.
So, आज we are doing the full translation mechanism, the genetic code, wobble hypothesis, ribosomal sites, energy costs, everything. Ready?
Ready?
चलो लेट्स गो. ओके मैम, पहले प्लेयर्स आइडेंटिफाई करता है. ट्रांसलेशन में कौन कौन इन्वॉल्व है?
Four main players. 1. mRNA, the blueprint, the instruction manual. 2. Ribosome, the factory floor, made of two subunits, small and large. 3. tRNA, the delivery trucks bringing amino acids. 4. Amino acids, the raw materials.
And the ribosome has three sides, correct? The A site, P site, and E site?
Perfect. Let me explain these because NEET loves asking this. A site, amino acyl site, where new tRNA arrives with amino acid. P site, peptidyl site, where the growing protein chain is held. E site, exit site, where empty tRNA leaves. Think of it like airport gates.
अरे एयरपोर्ट गेट्स. गेट ए पे नए लोग आ रहे. गेट पी पे लोग वेट कर रहे बोर्डिंग के लिए. गेट ई से लोग निकल रहे.
भाई दैट्स एक्चुअली परफेक्ट.
See, local references work. Now, mnemonic time. Arriving passengers exit daily. APE sequence. A site gets new tRNA, P site holds the chain, E site releases empty tRNA.
What about the genetic code itself? How does three nucleotides code for one amino acid?
The triplet code. Every three nucleotides on mRNA equals one codon equals codes for one amino acid. There are 64 possible codons. 4 cubed equals 64, but only 20 amino acids. So the code is degenerate. Multiple codons can code for the same amino acid.
वेट, तो इसका मतलब एक अमीनो एसिड के लिए मल्टीपल कोड्स हो सकता है.
Exactly. For example, leucine has 6 different codons, serine has 6, but methionine and tryptophan have only 1 each. This is the wobble hypothesis.
वॉबल मतलब लड़खड़ाना. क्या चक्कर है?
Kind of. The third position of the codon is wobbly. It can pair with multiple bases on the tRNA anti-codon. So one tRNA can recognize multiple codons. Francis Crick discovered this. Efficiency का चक्कर है.
So wobble allows for fewer tRNA types than there are codons. Makes sense from a cellular economy perspective.
Exactly. Only 61 codons code for amino acids. 3 are stop codons: UAA, UAG, UGA. Mnemonic: you are annoying, you are gone, you go away. These tell the ribosome to stop translation.
You are annoying. That's staying with me forever.
और स्टार्ट कोडॉन वो भी होता है ना?
Yes, AUG codes for methionine. Every protein starts with methionine in eukaryotes. It's the universal start signal. Think of it like good morning, conversation शुरू होता है इससे. Alright, translation का first phase, initiation. Prashansa, explain करो, how does the ribosome know where to start?
So in eukaryotes, the small ribosomal subunit binds to the 5' cap of mRNA and scans along until it finds the start codon, AUG. This is usually the first AUG after the 5' UTR region. The scanning model.
Perfect. But ये स्कैन करते करते कैसे पता चलता है कि यही वाला AUG सही है?
Because of the Kozak sequence, a consensus sequence around the AUG that signals start here. It's like context matters.
Exactly. Kozak sequence is like the sign that says assembly line starts here. Now, when the small subunit finds AUG, a special tRNA arrives. This is called initiator tRNA or met tRNA. It carries methionine.
तो ये special tRNA है? Normal tRNA से different?
Yes, it's the only tRNA that can enter the P site directly without going through A site first. VIP entry. And it needs help from initiation factors, proteins called EIF2, EIF3, EIF4. These help assemble everything.
इनिशिएशन फैक्टर्स मतलब स्टाफ मेंबर्स जो सेटअप करते हैं, जैसे इवेंट में ऑर्गेनाइजर्स?
Perfect. Event setup crew. They bring the small subunit, the met tRNA, and the mRNA together. Once AUG is positioned correctly with met tRNA in the P site, the large ribosomal subunit joins. Now the ribosome is complete and ready.
तो स्मॉल सब यूनिट पहले आता, स्कैन्स करता, AUG पे रुकता, मेट tRNA आता P साइट में, देन लार्ज सब यूनिट ज्वाइन करता.
Perfect sequence. Mnemonic. Small scans, met arrives, large locks. That's your initiation.
What about energy cost? GTP is hydrolyzed during initiation, right?
Yes, initiation uses GTP for energy. EIF2 needs GTP. This is a high-yield NEET point. Which molecule provides energy during initiation? Answer: GTP, not ATP.
GTP मतलब premium petrol? ATP regular petrol?
भाई ये एनालॉजी भी काम कर गया.
Honestly, yes. GTP is used for protein synthesis, ATP for other stuff. Now, prokaryotes do it slightly differently. They have a Shine-Dalgano sequence instead of 5' cap scanning. It's a specific sequence on mRNA that directly binds the ribosome.
So prokaryotes can start translation even before transcription is complete because they don't have a nucleus, right?
Exactly. Coupled transcription translation. But eukaryotes, transcription in nucleus, translation in cytoplasm, completely separate. Now comes the main event, elongation. This is where the protein chain actually grows. Arav, explain the cycle.
Okay, so ribosome is ready. Met tRNA is in P site. Next codon on mRNA is exposed at A site. A new tRNA arrives at A site carrying the next amino acid.
Correct. But how does the right tRNA arrive? Out of 61 types, how does it know?
Codon-anti-codon pairing?
Yes, the tRNA has an anti-codon. three bases that are complementary to the codon. If the codon is UUU, the anti-codon is AAA. Perfect match equals tRNA binds. This requires elongation factor EFTU and GTP.
And once the correct tRNA is in A site, peptide bond formation occurs between the amino acid in P site and the one in A site, catalyzed by peptidyl transferase in the ribosome.
Gold star. Peptidyl transferase is actually the ribosomal RNA. The large subunits RNRA acts as the enzyme. It's a ribozyme. NEET loves this. What catalyzes peptide bond formation? Answer: ribosomal RNA, not a protein.
अरे तो आरएनए एंजाइम बन सकता है? मतलब आरएनए भी मल्टीटास्किंग कर रहा?
Yes, RNA can be an enzyme. This won the Nobel Prize. Thomas Cech and Sidney Altman, 1989. Anyway, after the peptide bond forms, the chain is now attached to the tRNA in A site. P site tRNA is now empty.
तो अब क्या होता है?
Translocation. The ribosome moves 3 nucleotides forward on the mRNA. This shifts everything. A site tRNA with chain moves to P site site tRNA empty moves to E site. E site tRNA leaves. A site is now empty, ready for next tRNA.
And translocation requires EFG and another GTP hydrolysis, correct?
Exactly. So elongation uses 2 GTPs per cycle, one for tRNA binding, EF2, one for translocation, EFG. This is why protein synthesis is expensive energetically.
मतलब हर स्टेप में पैसा लग रहा है. जीटीपी जीटीपी जीटीपी. सेल का बजट खत्म हो जाएगा.
इतना मारूंगी ना.
इसलिए we need mitochondria, the powerhouse.
Exactly. Now this cycle repeats. New tRNA to A site, peptide bond, translocation, repeat, repeat, repeat. The chain grows amino acid by amino acid. Speed, about 15 to 20 amino acids per second in eukaryotes.
Wait, so a 300 amino acid protein takes only 15 seconds to make?
Roughly. Fast AF. And remember, multiple ribosomes can work on the same mRNA simultaneously. Polyribosomes or polysomes. Mass production.
ये तो हल्दीराम्स की असेंबली लाइन है. एक एक नामकीन पैकेट बन रहा कंटिन्यूअस.
Perfect analogy. Mnemonic for elongation. Binding, bonding, translocation. 3 steps, repeat करो.
Okay, प्रोटीन बन रहा है कंटिन्यूसली. कब रुकता है?
अब स्टॉप कोडॉन ए साइट पे आ जाता है. UAA, UAG या UGA. Remember, you are annoying, you are gone, you go away.
And there's no tRNA for stop codons, right? Instead, release factors bind?
Perfect. No tRNA exists for stop codons. Instead, release factors, ERF1 in eukaryotes, recognize the stop codon and bind to A site. This triggers the ribosome to release the completed protein chain.
तो रिलीज फैक्टर बेसिकली बाउंसर की तरह है. पार्टी ओवर, एवरीवन आउट.
Yes, the peptidyl transferase adds a water molecule instead of an amino acid. This breaks the bond between the protein and the tRNA. Protein is released. Then the ribosome dissociates into small and large subunits. Everything falls apart. Job done.
मतलब दिवाली में पटाखा जलाया, धूम धड़ाका हुआ और फिर सब चले गए.
That's one way to describe it.
Now, NEET traps for termination. 1. How many stop codons? 3. UAA, UAG, UGA. 2. Do stop codons have tRNA? No. Release factors bind instead. 3. What happens to the ribosome after termination? Dissociates into subunits, can be reused.
Recycling system hai basically.
Exactly. Ribosomes are reusable. One ribosome can make thousands of proteins. Now, energy cost for entire translation? Initiation, 1 GTP. Elongation, 2 GTPs per amino acid. Termination, 1 GTP. For a 100 amino acid protein, roughly 200 plus GTPs.
That's incredibly energy intensive. No wonder defects in translation cause major diseases.
Yes, like cystic fibrosis, premature stop codon in CFTR gene. Translation stops early, protein is incomplete, disease happens. Or antibiotics like streptomycin. They block bacterial ribosomes, stop translation, kill bacteria.
अरे तो एंटीबायोटिक्स बैक्टीरिया का ट्रांसलेशन रोक देते हैं, मतलब उनका प्रोटीन फैक्ट्री बंद कर देते हैं।
Exactly. That's how many antibiotics work. They target prokaryotic ribosomes, 70S, but don't affect eukaryotic ribosomes, 80S. So bacteria die, you survive.
भाई दैट्स जीनियस।
Alright, final recap. Ma'am, translation in 30 seconds.
Done. Initiation. Small ribosomal subunit binds mRNA 5' cap, scans for AUG start codon. Met tRNA enters P site. Large subunit joins. Ribosome complete. Elongation. tRNA with amino acid enters A site. EFTU GTP. Peptide bond forms ribosomal RNA catalyst. Ribosome translocates 3 nucleotides EFG, GTP. Repeat, chain grows. Termination, stop codon. UAA, UAG, UGA reaches A site. Release factor binds, protein released. Ribosome dissociates. Finished.